All right, so we're going to move on to our educational presentations, and we're going to start with Jeremy Sackett from Cascadia Labs. So I'd like to welcome Jeremy up to the stage.
Well, good evening, everybody. I am Jeremy Sackett with Cascadia Labs, based in Bend and Portland, Oregon. Thank you very much, Bethany and OG Analytical, for inviting us down today. We're going to talk a little bit about Potency. Bethany already stole a little bit of the thunder with the myth busting of Potency with the trivia. I was going to start with about the same question, what everybody thought an average potency was, but we already got through that, so we'll just move on. We are going to talk a little bit about purity, potency, and then how that relates to analytical chemistry and the analysis of cannabis and cannabinoid products. Coming from a pharmaceutical background primarily, in my previous experience, look at purity and potency as two unique items, and I think just defining what those are will be helpful just as a general starter here tonight. So looking at purity. Purity is essentially as it's defined by the Code of Federal Regulations, relative freedom from extraneous matter in the finished product. So what does that really mean? That is high purity drug products, or drug substances primarily. Coming from a pharmaceutical background, the active ingredient would be of high purity and then formulated into a drug product with other excipients. This is very different than what we find obviously in medical cannabis or other botanical-based medical products that are available. Looking at purity standpoint, though, this could be applied, and it is applied to high purity delta-9 THC or other high purity extracts of cannabis plant material. When you're reaching above 95% of a certain ingredient, you're really starting to look more at a purity than likely an overall potency of that product under test. So potency. Potency is really looking at the ability of a product to enact a specific response or a desired response of those compounds. For the amount of drug product that is necessary to achieve a desired effect. So thinking of cannabis and delta-9 THC, one of the noted effects, is euphoria. And it's just as a side note, it's really interesting. The Journal of American Medical Association has put out a few recent clinical trials and just kind of looking at evaluations of cannabis-based products. And one of the adverse effects is euphoria. I just thought that was kind of a unique adverse effect. I personally like euphoria. But looking at that effect, and that effect of euphoria, or the effect of nausea, a reduction of nausea, or increase of appetite, there's a certain amount of cannabinoids that need to be delivered to the body to achieve that effect. And that is what the potency of that product is. You'll hear a lot about practices or rules that are coming out where you're looking at 5 milligram dosing limits, 10 milligram dosing requirements. A lot of those limits are based on the known effect of those cannabinoids to alter perception or alter the user experience. So just looking at kind of the dose response curve and how those limits would hopefully be defined, using data and science to drive what those limits would be, is you would look at product A here on this dose response curve. Down here is the concentration of the active ingredient, here being the response or the user effect. Product A having a lower concentration to achieve this same effect would be more potent than product D here. So that would be, for instance, if product A were formulated, a cannabinoid product that was more bioavailable. For instance, if I was going to smoke and have an inhalation of delta-9 THC, that would deliver more of that to my body than if I were to eat the same amount of that delta-9 THC. So a product with the same amount of that active ingredient could be more potent, dependent on how that is consumed. Okay, so how does this why does this matter for a cannabis science pub? This is really looking at potency, is how we're reporting potency from the cannabis-centric analytical laboratories, and how consumers are using potency values to make decisions on the products that they are going to consume on a day-to-day basis, or whatever cycle they choose to consume those products. So we look at solids and liquids primarily as how we're going to break down how potency is reported for cannabis products. Potency for solids is typically conveyed as a mass fraction, and that will be the milligrams of THC per milligrams of product. And most often that is multiplied by 100 to give us the percentage by mass. And this is the most common format for flower and solid extracts: 13%, 18% for a flower, 65%, so on and so forth for an extract. And what that actually means is the milligrams of the active ingredient, delta-9 THC in this case, over the total weight of that product. And that's going to be the same representation of potency that would be used for an edible product. It is just represented slightly different for edibles because of the consumption mechanisms that we use. Most folks are used to the percentage value for flowers, but when you look at an edible product, we're still looking at milligrams of active ingredient for the total product. For instance, this cookie will have 25 milligrams of THC. When looking at solids, it's always the milligrams of active, or a weight of active could be grams per grams of the total product. So with liquids, this is where we've had a lot of inquiries to the laboratory on, what does milligrams per milliliter mean? How can I relate this tincture to my flower? How can I relate this edible to my tincture? And it's really, I think, for the cannabis consumer to really start looking at the milligrams of delivery and start shifting away from the percentage, particularly for solid products, or just the understanding that you can always get back to the milligrams of active ingredient that you're delivering, and hopefully we come up with a labeling system in new rule promulgation here in Oregon, nationwide, that will convey that information appropriately to the general public. What we're looking at for for liquid products is the milligrams of that active ingredient per volume of the total product. So that would again be milligrams of THC per milliliter, and the way that those products would be dosed is by a volume. For instance, 1 mLs would be 1 milligram of THC. As I alluded to a little bit, percentage by volume is often not appropriate for liquid tinctures or other liquid products on the marketplace because they are a much lower percentage by mass percentage, or percentage by volume, comparable to the percentage by weight. So a consumer could see a 0.01 or a 0.1% tincture and think, oh, it's quite low, I can drink the bottle, and that is obviously going to not be ideal effects. So kind of getting into laboratory testing of cannabis potency, really just trying to explain some of the steps in the procedure. This is consistent for most analytical sciences, where there's, in my opinion, 3 steps. It's not a one-size-fits-all method for potency for any analyte. It's really the sample preparation is the critical step, in my opinion. These other instrument analysis and data interpretation are very, very important steps. But as in the context of our discussion tonight, we're really looking at the challenges of sample preparation and extraction, and how the different cannabis products present those challenges. It must be suitable for the product. For instance, drug products have methods that are developed uniquely for that product. So lisinopril has its own methods dedicated to evaluating the potency of that lisinopril product. So when new cannabis products come on market that are unique formulations in a medical product, perhaps have polymers or other additives that will enhance bioavailability, I really see these products coming to market sooner than we may realize. It's going to be important that you can't just send a sample to a laboratory and expect them to be able to analyze the full suite of services that they offer. For instance, we just received bacon the other day, cannabis-infused bacon, and we're requested to do a pesticide analysis on it. It's very challenging to perform an analysis on a unique matrix, and that's where we're going to go next, is the complexity of the matrix is really driving the challenges here. I could say, yes, we can do a dilute and shoot, run your bacon, but I'm not going to have valid data to report. And that's where, as we move into accreditation, it's not just throw it on the machine and see what comes out. It's going to take time to look at what the matrix is, what the analyte of interest is, and what the technology that we're using. And that is going to be the scope of accreditation. I have a slide just after this that will talk about a little bit those aspects. But the diversity of even just the biological plant matrix here, not even talking about formulated products with unique additives or excipients, but just the plant material itself is so complex from an analytical standpoint. The number of chemicals, phytochemicals in that matrix that we need to separate and then measure each one independently is sometimes mind-boggling. And the real challenge is the extraction efficiency, ensuring that you're getting out all of the cannabinoids and leaving behind the plant material and other extraneous, non-important materials. The separation of all of those cannabinoids, ensuring that your terpenes and cannabinoids are not coming out at the same time. The workflow demands. From an analytical standpoint, I could run a method for 2 hours on, a 120-meter column and separate everything very easily. But from practicality standpoint, we can't take, several hours for one sample. We have to try to keep those methods short so that we can get meaningful data out, but still separate all of those. So, I feel that these are the primary challenges on a day-to-day basis that we face, analyzing this complex matrix. And I did speak to the addition of various ingredients to create these new products, or even just infused products with challenging gummy matrices or different baked goods and different sugars, other things that will hold on to those cannabinoids in a different manner than the product next to it, speaking to extraction efficiency. So now getting into the matrix analyte here, this is all under the TNI standard in ORLAP, which was we were discussing a little bit with the updates earlier. The TNI standard is the standard that the laboratories will be operating under. An ORLAP there is the Oregon Environmental Laboratory Accreditation Program. That is the group out of the Oregon Health Authority that will be inspecting laboratories to ensure that for every product type that we're charged with analyzing a potency value for, we have a separate scope. So as laboratories become accredited, it is possible that a laboratory will only be testing potency for flower. That would be the biological matrix. A laboratory could choose not to be accredited for the solid and chemical matrix of edible and extract. And that is where, as we move forward, laboratories are either going to choose a full, to undergo accreditation into all of these different scopes of accreditation. It's not just saying, I'm going to be accredited as a cannabis laboratory. You need to identify that you are going to test Delta-9 THC, THC, so on and so forth, pesticides in plant material using LC triple quad. And you have to do this for each of the different analytes and each of the different matrices that you are going to become accredited for. And that is pretty significant, and I think that is going to change really the operations and the data valid data that we have available to really ensure consumer safety through knowledgeable information on the labels. And I think that's where we really get to what we're trying to do here as an analytical community in Oregon is really trying to develop sound practices, set the standard, and have a successful industry with good potency information available for the consumer. And I'll leave it at that, I guess. Thank you all for your time. Any questions?
Let me bring you the microphone. Oh, that's really loud.
Do you think that the Oregon certification program will include a separation between plant matrices and edibles?
Absolutely,
Is this
100%.
Definitely an important differentiation for extraction.
Yes, 100%. These matrices here, biological matrices, solid and chemical, and water, are the three matrices identified by the TNI standard, and this is how they will likely fall out for cannabis with those being there. Tinctures and liquid preparations are still kind of being determined. There's a rules advisory committee and adjustments to the actual rules for ORLAP right now. So there's several kind of administrative rule changes that are ongoing to support this industry. ORLAP has done a phenomenal job to consider all the different product types that are actually in the cannabis industry, and they are looking at how to best address those. But yes, definitely Cannabis Laboratory accreditation will include multiple matrices as well as analytes, obviously.
Per the water matrices, are they going to be requiring acetate to be tested then, or?
I can't comment on that. I don't believe so. I don't know.
So will there be tests for the quality of the fats, like you're talking about bacon fat versus like polysaccharide type of fats or monosaturated type of fats, etc.? I mean, I would think there'd be a difference in the potency of those, because a lot of the THC is absorbed in the adipose tissues, right? It's stored in our adipose tissues.
Right. So I think, please correct me if I'm wrong, but the question being, looking at the different types of fats, they would be obviously different matrices. They would be included in the biological matrices, one category per the TNI standard. It is my understanding that through the inspection process, you would identify, more specifically in your methodology, these types of fats, and then through the review of your validation data, either pulled directly from a standardized method or verified internally that you can meet, those requirements for those unique. I think the difference between those types of fats would be contained within the method validation data package, then reviewed by ORLAP and approved through that mechanism. But my understanding is they would be within the same scope of matrices. Did I answer your question? I'm sorry if I didn't, if I misunderstood it.